Right. In the previous lectures, we talked in detail about the insulin synthesis in the beta cells, insulin storage, and also insulin secretion and other factors that play a role in regulating of insulin secretion. Today, we are talking about another topic, which is insulin receptors and signaling pathways. Insulin receptors and signal transduction. Okay. This lecture is only a basic introduction into the insulin signaling pathway. In the next lectures, we talk about about the insulin actions in target tissue, in target cells, in detail. Okay, you know that insulin is a very powerful anabolic hormone, and insulin is an anabolic hormone. What do you mean by anabolic? Anabolic hormones, or generally anabolism or anabolic pathways, are these reactions which lead to the formation of large and complex molecules from the smaller ones. Anabolism is formation of large and complex molecules from smaller ones. For example, glucose may be converted into what? Into glycogen. Glycogen. This is an anabolic reaction. Glucose is a small molecule, is a simple sugar, and converted into glycogen, which is a long branched chain of glucose. And this conversion of glucose into glycogen is called an anabolic reaction. Also, amino acids may be converted into peptides or proteins. Peptide or proteins. Again, conversion of the amino acids into peptides and proteins is an anabolic reaction. And also, fatty acids are converted into what? Fatty acids are converted into triglycerides or other larger lipids. And this reaction again is an anabolic hormone because it goes through an anabolic pathway, which is conversion of small molecules into complex and large ones. And actually, insulin is not an anabolic hormone, it's also anti-catabolic. What's anti-catabolic? It's anti-catabolic hormone. Anti-catabolic is just opposite to anabolic action. Catabolism means breaking down of this glycogen back into glucose, and this is a catabolic reaction. Again, protein breakdown into amino acids by protease enzymes is a catabolic reaction, and breaking down of triglycerides into fatty acid and glycerol is again another catabolic reaction. So insulin is an anabolic hormone and anti-catabolic hormone. Why it's important? Because insulin secretion from the beta cells is associated with energy abundance. When there is high amount of energy, high amount of glucose, high amount of amino acids or fatty acids, insulin in the beta cells are stimulated and secrete insulin into the blood. And in turn, this insulin leads to proper utilization of these nutrients and storage of excess nutrients by target tissues. And what are the target tissues for insulin? The target tissues generally are, or the classical targets for insulin, the first one is what? The liver. Okay? The liver is one of the classical targets of insulin, the muscle tissue, and also the adipose tissue. The liver tissue, muscle cells, and adipose cells are the classical targets for insulin action. Also, insulin receptors are present in almost all the other cells. We can talk about the other action of insulin in other tissues later, but first of all, we focusing on the insulin signal transduction in the liver, in the muscle, and in the adipose tissue, and then we talk about the insulin action in this tissue. So let's start it with insulin receptor. You know, insulin is a peptide hormone, and it's composed of what? Of two peptide chains, the A chain and the B chain, which are linked by disulfide bonds. And it's about 51 amino acids. Insulin is 51 amino acids. Because 51 amino acids is a large peptide, it cannot penetrate the plasma membrane in the cell, so it needs to transmit its signal through a receptor. Let's suppose this is a target cell for the insulin. Maybe this is a cell in the muscle, in the liver, or any other cells. And insulin cannot penetrate this plasma membrane, so it will bind and activate a cell surface receptor to exert its action. Here we have insulin receptors. Insulin receptors is composed of 4 subunits, 4 peptides, 2 beta-ons, and 2 alpha-ons. These are the 2 beta-ons, 2 beta subunits, and also we have 2 alpha subunits. Okay? Insulin receptor is composed of 2 beta subunits and also 2 alpha subunits. This is the beta-on and this is the alpha subunit. The beta subunit is linked to the alpha subunit through a disulfide linkage, and the 2 alpha subunits are again linked to each other by the disulfide bond. This is the insulin receptor, which is composed of 2 beta subunits and also 2 alpha subunits. So it's called heterotetrameric receptor. It's between the 2 alpha subunits, there is a domain which is specific for binding of insulin. First of all, insulin is come to here and bind to somewhere between these 2 domains. This is the insulin and binds to somewhere between these 2 alpha subunits. Once the insulin is bind to this alpha subunit, a conformational change occurs in the whole receptor, and the beta subunit, which is a transmembrane peptide, the alpha subunit is completely outside the cell. The beta subunit is transmembrane, means part of it is outside the cell, part of it is transmembrane inside the plasma membrane, and part of it is intracellular. In the intracellular part of the beta subunit, there's a special domain. This domain has enzymatic activity. Once the insulin is bind to the alpha subunit, the beta subunit is activated, and this enzyme will phosphorylate the beta subunit. This enzyme will phosphorylate the other one, adding phosphate groups into the other beta subunit, and this one phosphorylate the other beta subunits. So what will happen? Phosphate groups are added into the beta subunits. It's the phosphate groups on which residue? On the tyrosine residue. So this enzyme is called tyrosine kinase, because it is only adds phosphate groups into the tyrosine residues on the beta cells or other target proteins. And you know what's kinase? Kinase is any enzyme which adding phosphate groups to other proteins. Enzymes or proteins generally we have in 2 states. Let's suppose this is an enzyme. Okay, this is an enzyme, and here is the active site for acting on other proteins. This enzyme may be active when it is phosphorylated. When adding a phosphate into this enzyme will be active, while some enzymes are active when there is no phosphate groups. Adding a phosphate group into an enzyme or any other protein or any other molecules is called what? Phosphorylation. So if you ask what's phosphorylation, phosphorylation is the process of adding a phosphate group into another molecule, mostly proteins or enzymes. And removing this phosphate from this enzyme is called what? Dephosphorylation. So we have some enzymes which are active when it's phosphorylated, while other enzymes are active when they are not phosphorylated. Some of them are active when it's phosphorylated, while others are active only when not phosphorylated. And it largely depends on the enzyme structure, on the protein structure, on the amino acid sequence, and also which amino acid is phosphorylated. The phosphate group is added to which of the amino acids, maybe serine amino acid, threonine, or tyrosine. In this case, this tyrosine kinase is adding phosphate groups into the beta subunit, and it's called what? Autophosphorylation, because it's phosphorylated by a special domain in the same peptide, or sometimes called transphosphorylation, because this tyrosine kinase phosphorylates the other beta subunit, and this tyrosine kinase phosphorylates the other beta subunit. An insulin will never enter the cell. It's only bound to its receptor, and activation of the receptor leads to a series of reactions, a cascade of reactions of phosphorylation, dephosphorylation, and also second messenger generation, and protein interactions. By changing phosphorylation, dephosphorylation, formation of the second messengers, and protein-protein interactions, insulin exerts its action on the target cells and leads to altering the function of the membrane, changing the membrane permeability to amino acids or other molecules, also changing the enzymatic activity, the enzymatic machinery in the cytosol, and even the insulin is alter the nucleus. Gene transcription of the cell may be altered by the insulin action. So by binding of insulin to its receptor, changes in the membrane, in the cytosol, and also in the nucleus will occur. After activation of insulin receptor by phosphorylating it from the tyrosine residues by the tyrosine kinase, other proteins will come to the receptor. For example, one of these proteins is this protein, which binds to this area of the receptor which is phosphorylated, and this protein now is active. What's the name for this protein? It's called insulin receptor substrate, and you have type 1, 2, 3, 4. Insulin receptor substrate IRS type 1 to 4 is bound to the insulin receptor when the receptor is activated by phosphorylation. And once this protein, this IRS, is bound to the receptor, it's also phosphorylated by the tyrosine kinase of the beta subunit. Phosphate groups are added to this IRS. Here are the phosphate groups added to this IRS. IRS has no enzymatic activity, but it only acts as an adapter protein, links the insulin receptor to other proteins in the cytosol. One of these proteins will bind to this IRS as a protein or an enzyme, come to here and bind to it. Okay, this enzyme will come to the IRS and bind to it. So the IRS is act as what? As an adapter protein, because it only joins the insulin receptor to other enzymes and target proteins. Once this protein or enzyme is linked to the IRS, it will act on the plasma membrane by converting what? Membrane phospholipid is here, which is called phosphatidyl inositol. Phosphatidyl inositol bisphosphate is constitutively on the plasma membrane, and this phosphatidyl inositol, or simply called PIP2, is affected by this enzyme and converted into what? Into phosphatidyl inositol 3-phosphate. PIP2 is phosphorylated on 2 carbons, but now it's phosphorylated at 3 points. 1, 2, 3. PIP2 is phosphatidylate inositol bisphosphate, phosphorylated in the 4 and 5 members of the ring, while the PIP3 phosphatidylate inositol triphosphate is phosphorylated in 3 members of this ring. PIP2 and PIP3 are membrane phospholipids, and this enzyme, which is responsible for the conversion of PIP2 into PIP3 by adding what? By adding a phosphate group from the ATP, will activate this PIP3. Now the PIP3 has more negatively hit and protruding into the cytosol. But what's the name for this enzyme? It's called PI3 kinase. PI3 kinase, okay? Because it converts PIP2 into PIP3. After conversion of the PIP2 to PIP3, which is a membrane phospholipid, other proteins will come to here and they will bind to the PIP3. One of them is called what? PDK. PDK means PIP3 dependent protein kinase. PIP3 dependent protein kinase, and it's maybe type 1 or type 2. Another protein will come to here. This protein is named protein kinase B, or simply ACT. Protein kinase B or ACT, AK straight transforming protein. When these two proteins, which are kinase enzymes, bind to the PIP3, the PDK 1 or 2 will be activated and phosphorylate the protein kinase B. This protein kinase B is now phosphorylated, which is some proteins or enzymes are active when they are phosphorylated, and the others are only active when non-phosphorylated. The protein kinase B is from these proteins which are active when it is phosphorylated. Adding a phosphate to it, and then it will be activated. PDK will activate the protein kinase B by phosphorylation, and also it activate another protein kinase, which is called protein kinase C. This is the protein kinase C, type lambda or zeta. Then protein kinase C is again active when it is phosphorylated. Okay. Now again, the insulin bind to its receptor between the alpha subunits, and this binding of insulin receptor leads to a conformational change in the receptor shape and leads to the activation of the tyrosine kinase. Tyrosine kinase leads to the phosphorylation of the beta subunit, and then the IRS is bind to the receptor and is again phosphorylated by the tyrosine kinase. Once the IRS is phosphorylated, it will recruit other proteins from the cytosol. One of them is PI3 kinase, which converts PIP2 into PIP3. PIP3 is now active and bind to two other enzymes, which are the phosphatidyl PDK, PIP3 dependent kinase, and also protein kinase B. Also, PDK will activate and phosphorylate the protein kinase C. Now we have two active enzymes. One of them is protein kinase B, and the other one is protein kinase C. What will the function of these enzymes? Both goes to the cytosol and acting on other enzymes. The protein kinase B modulates the function of some other enzymes, and again the protein kinase C modulate the function of other enzymes or proteins, which are called motor proteins. Motor proteins are proteins help for the movement of special vesicles in the cytosol, which loaded with what? Loaded with GLUT4. Here we have a special vesicle. This vesicle is loaded with GLUT4, glucose transporter type 4. Okay, this is the glucose transporter type 4. Okay, we have many GLUT4s on the membrane of these vesicles, and they are called GLUT4 vesicles. GLUT4 vesicles. By altering the other proteins, motor proteins, which are here, this vesicle will be more motile and translocated into the plasma membrane and leads to the fusion of this vesicle with the plasma membrane and inserting these GLUT4s into the plasma membrane. Now, this vesicle, this vesicle is fused with what? With the plasma membrane, which contains many GLUT4 carrier proteins. What are the functions of these GLUT4s? Of course, transporting glucose from the outside of the cell to the inside of the cell. Okay, when the insulin is come to these cells, when there is high amount of glucose, this glucose, because it cannot directly diffuse it to the cell, it needs a carrier protein to be transported to the inside, and insulin from the PIP3 and protein kinase B pathway changes the function of motor proteins that leads to the translocation and fusion of the this GLUT4 vesicles and leads to fusion of this GLUT4 into the plasma membrane. Now the glucose enters the cell, right? And we have more glucose inside the cell. This is the glucose, okay? This glucose is then converted into what? Into glucose or 6-phosphate. Glucose, after entering the cell, it will be converted into glucose 6-phosphate by the action of an enzyme which is called hexokinase. What's the function of the hexokinase? Hexokinase is responsible for the conversion of glucose into glucose 6-phosphate by adding a phosphate group to the carbon number 6 of the glucose. This glucose 6-phosphate may enter the glycolysis or other pathways for production of what? For the production of ATP, right? It will produce ATP, and this ATP is used for what? For supplying the energy for the cell, or the excess amount of the glucose, which is the form of the glucose 6-phosphate, may be converted into glycogen to be stored for future needs. Glycogen. So, insulin leads to increasing uptake of cells through inserting more and more GLUT4 into the plasma membrane, and when more glucose enters the cell, glucose is directly converted by the hexokinase enzyme into glucose 6-phosphate, and this glucose 6-phosphate may be used for production of ATP, and this ATP supplying the energy needs of the cell, or it may be stored in the form of glycogen. Okay? In which cell we have GLUT4? When you talk about GLUT4, we should know that GLUT4 are present in the muscle tissue and also in the fat tissue. We don't have GLUT4 in the liver. Insulin also increases the uptake of glucose by liver cells, but it has another mechanism that you can mention it later. But insulin increases glucose uptake by the skeletal muscle and also by the peripheral adipose tissue by increasing the number of the GLUT4 on the plasma membrane. Now, let's come to another function or other functions of the protein kinase B. Okay, about the other functions of the ACT protein. In humans, we have ACT1, ACT2, and ACT3. Usually it's ACT2, which is involved in the insulin signaling pathway, or protein kinase B type beta. Okay, the ACT2 now goes to targeting other proteins and enzymes in the cytosol. One of these enzymes is what? Is mTOR. Okay, ACT targeting the mTOR. What's mTOR? It's a very important enzyme which is called mTOR. mTOR stands for mammalian target of rapamycin. Mammalian target of rapamycin. Rapamycin is an immunosuppressive agent. First it was found without knowing its target, and then it was found that rapamycin will bind to this protein and it's called mammalian target of rapamycin. Rapamycin, or also called sirolimus, is an immunosuppressive agent used in many conditions, for example, in prophylaxis for renal transplant rejection. Okay, ACT will phosphorylate the mTOR on serine or threonine residues. So now this mTOR is phosphorylated and it is active. Again, ACT is also phosphorylated on serine and threonine residues. The first tyrosine which we have was, the serine kinase which we had was what? Tyrosine kinase. The rest of the kinases all have serine threonine kinase activity. ACT is a serine threonine kinase. mTOR is another serine threonine kinase. Okay. And what is PI3 kinase? It is tyrosine kinase or serine and threonine kinase. It's not a protein kinase, it's lipid kinase. PIP2 is a phospholipid, membrane phospholipid. It's not protein kinase. So, the ACT will phosphorylate the mTOR, and now mTOR is active. mTOR activate another protein kinase, which is called what? S6K. This is the S6K. S6K stands for the ribosomal protein S6, and this is the S6 kinase. Okay, and also this S6 kinase is active when it is phosphorylated. It's fully functional when phosphorylated, and without phosphorylation it is inactive. Okay, this S6 kinase will activate another protein called what? PP1. This is the PP1. PP1 is protein phosphatase type 1. It means it's not a protein kinase, it is phosphatate, removing phosphate from other enzymes or proteins. It's active when phosphorylated by what? By S6K. And what's the function of the PP1? PP1 acting on two important enzymes for the regulation of the metabolism of glycogen. The first one is what? The first one is glycogen synthase. Glycogen synthase, or simply GS. Glycogen synthase is an enzyme responsible for the formation of glycogen. Glycogen is a long chain, branch chain of glucose, repeated units of glucose. And this glycogen synthase is responsible for adding more and more glucose to this glycogen. For example, this is a glycogen. Okay. And this glycogen synthase will come to here. Glycogen synthase will come to this glycogen and adding other glucose to it. Adding glucose, for example, here, glucose, 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 and then more glucose and more, until this complex will become a large branched chain of glycogen. So glycogen synthase is active in dephosphorylated state. When it's not phosphorylated, it's active and able to adding glucose into the glycogen. But if we adding a phosphate group to this glycogen synthase, let's suppose this phosphate group added to this glycogen synthase, what will happen? It will be inactive. So this protein phosphatase 1 remove any phosphate groups on the glycogen synthase. And the glycogen synthase is now ready to act on the glycogen for adding more glucose units. So the PP1 is activating the glycogen synthase. Again, this PP1 acting on another enzyme, which is called what? Glycogen phosphorylase, not synthase. This is the glycogen phosphorylase. The action of the glycogen phosphorylase is opposite to the glycogen synthase. Glycogen phosphorylase goes to the glycogen and removing the glucose. Remove this glucose and then this one and this one until breaking down all the glycogen. So because insulin is an anabolic hormone and wants to form more glycogen from the glucose into the cell, it will stimulate the glycogen synthase to increase the formation of glycogen, or simply we call it glycogenesis. And also at the same time, insulin is an anti-catabolic hormone. It means inhibiting the glycogen phosphorylase and inhibiting the glycogen breaking down. By what? By activating the PP1, protein phosphatase 1, which is responsible for removing any phosphate groups on the glycogen synthase and removing glycogen phosphorylase, because it is inactive when dephosphorylated. Okay? Phosphate group is removed from the glycogen synthase and now it's inactive. And phosphate groups are removed from the glycogen synthase. Now it is what? Active. The ACT protein affecting the mTOR by direct phosphorylation. It also activating it by indirect phosphorylation. How? Here we have another protein which is called RIP. RIP stands for what? RIP is RAS homology enriched in brain. It's similar to the RAS protein, but it's highly found in the brain cells. RIP is active when it is bound to what? It's bound to GTP, guanosine triphosphate. This is a GTP bound to this RIP, and now it's active. When the GTP is bound to the RIP, it will activating the mTOR by not enzymatic action, by protein-protein interaction. But this RIP usually converted or removed the GTP into what? Into GDP by the action of another protein. This RIP is converted into what? Into the GDP form. And this is the GDP. You know that GDP is in low energy and the RIP is inactive. This form is inactive, which is bound to the GDP, but the GDP form is active and able to activating more strongly the mTOR, mammalian target of rapamycin, or sometimes called mechanistic target of rapamycin. Who converts the GTP or exchanges the GTP for GDP is another protein, which is here and called TSC1. TSC1 or TSC2. TSC is what? Is tuberous sclerosis complex 1 or tuberous sclerosis complex 2. Because mutation in the gene that encodes for this protein is linked to tuberous sclerosis syndrome, which is a rare multigenetic syndrome affecting many organs, including the brain, heart, or the kidney. It's called tuberous sclerosis complex 1. Tuberous sclerosis complex 1, or simply TSC1 or TSC2, are able to converting GTP or exchanging the GTP into GDP. It's in a group of proteins which is called GAP. GAP means what? GAP means guanosine or GTPase activating protein. This is the GAP. GAP means GTPase activating protein because it activating the intrinsic GTPase activity of the receptor. Here, for example, in the rib, we have a special domain responsible for GTPase activity, removing the GTP from the protein and adding a GDP instead of the GTP. The GAP or the TSC1 or TSC2, which are belongs to the family of GAP proteins, will come to here, come to this domain, and activating this domain, which is GTPase enzyme, by at least 1000 folds. This what? This is the GTPase. GTPase is now activated by at least 1000 fold and removing what? It will removing the GTP for what? For GDP. What's the role for the ACT protein? Simply, the ACT will come to this TSC1 or TSC2 and block its action. How? By phosphorylating it. Adding phosphate groups to this TSC2 in the serine or threonine residues, it will be what? Inactive. And then when it's inactive, it will not able to affecting the GTPase activity of the rib protein. So the GTP will remain bound to the rib, and the rib goes to activating the mTOR, mammalian target of rapamycin. In this way, by activating the mTOR enzyme and then S6 and PP1 enzymes, insulin alters the activity of the glycogen synthase and also glycogen phosphorylase to alter the metabolism of the glycogen, increasing glycogenesis, formation of the glycogen, and also inhibiting the breaking down of what? Glycogen. Let's go to another function of the ACT protein. It also activate another protein which is called what? Called or enzyme phosphodiesterase. Phosphodiesterase, when activated, converting what? Converting cyclic AMP. Cyclic AMP into what? Into AMP. Cyclic AMP is responsible for the activation of another enzyme, another kinase enzyme, which is called protein kinase A. Okay? Protein kinase A is active only in the presence of high amount of what? Of cyclic AMP. Or sometimes it's called cyclic AMP dependent protein kinase, because its activity is only depends on the amount or concentration of the cyclic AMP. Protein kinase A in turn is responsible for altering the function of many other enzymes. For example, it will activate the glycogen synthase. No, it's phosphorylate it and inhibiting it. And also it will phosphorylate the glycogen phosphorylase and activating it. So the protein kinase A is responsible for breaking down of glycogen and inhibiting its formation or formation of new glycogen. Insulin is an anabolic hormone and don't want this protein kinase A to be active. So in this way, it will inhibit the protein kinase A by breaking down cyclic AMP. Protein kinase A will not be active. Also, protein kinase A is responsible to altering the function of other enzymes, contributing to lipid metabolism. For example, it will phosphorylate an enzyme which is called hormone sensitive lipase, or this hormone sensitive lipase or other adipose tissue triglyceride lipases, and when they are phosphorylated by the protein kinase A, this will be more or less effective. Hormone sensitive lipase will be more effective and contributing to what? To lipolysis. Lipolysis. Lipolysis means breaking down of triglycerides into fatty acids and glycerol, and insulin wants to store fatty acids instead of breaking down of triglycerides into fatty acids, because already the cell is rich in fatty acids. Also, protein kinase A is responsible for phosphorylating and thus inactivating two other enzymes. Okay, two other enzymes are affected by the protein kinase A. One of them is ATP citrate lyase, ACL, and the other one is ACC, acetyl-CoA carboxylase. Both enzymes are responsible for what? For formation of fatty acid from acetyl-CoA. So you can say lipogenesis. So protein kinase A, which is its activity depends on the cyclic AMP, leads to activation of the hormone sensitive lipase and thus breaking down of triglycerides into fatty acids and inhibiting formation of new fatty acids. But the insulin through the ACT pathway leads to the activation of the phosphodiesterase E, and this will converting cyclic AMP, breaking cyclic AMP into AMP, and the protein kinase A will not be more active. So in this way, insulin increase lipogenesis and inhibit lipolysis, right? One more function of the ACT protein is what? It will affecting another protein which is named PGC1 alpha. What's PGC1 alpha? PGC1 alpha stands for pyroxene proliferator activated receptor gamma coactivator 1 alpha. Pyroxene proliferator activated receptor gamma coactivator 1 alpha. And this responsible for what? Here is the nucleus. In the nucleus we have what? We have DNA, right? And this is the DNA. In the nucleus we have a transcriptional factor which is called FOXO1. FOXO1 is only active in the presence of PGC1 alpha. PGC1 alpha will bind to the FOXO1 and both alter the transcription rate for a set of genes. Numerous genes are affected by the insulin. Some of these genes are responsible for metabolism and others are responsible for apoptosis. Insulin, by acting the protein ACT, which is protein kinase B, inhibiting the PGC1. This PGC1 is what? Inhibited, and it will not activate in the FOXO1. So the transcription of these genes will be inhibited. Why insulin wants to inhibit the transcription of these genes? Because the FOXO1 is responsible for two sets of genes. The first one are genes regulating the gluconeogenesis. Gluconeogenesis is formation of glucose from non-carbohydrate sources, for example from amino acids. And insulin don't want us to breaking down amino acids for the formation of glucose, because already we are rich in glucose and other nutrients. And also the other set of genes are called what? Bad genes. Bad genes, which are actually they are bad because it's responsible for what? Apoptosis. Apoptosis means program cell death. So the insulin through the ACET pathway leads to what? To inhibition of the PGC1 alpha, and then focus on, and then the gene in the cell in the nucleus will not be transcripted. Their genes include what? The genes for gluconeogenesis enzymes, for example, polyenol pyruvate carboxykinase, and also the bad genes group, which are responsible for apoptosis. One more thing that I want to mention it is an enzyme is very important, which is called glycogen synthase kinase type 3 beta. Glycogen synthase kinase 3 beta is a kinase enzyme. It's a serine threonine kinase enzyme again, acting on the glycogen synthase and also glycogen phosphorylase. By adding phosphate groups to glycogen phosphorylase will be what? Active. Okay. By adding phosphate group to the glycogen phosphorylase, activate it, and at the same time, adding phosphate groups to the glycogen synthase and inactivating it. The point is, the ACT protein will phosphorylate the glycogen synthase kinase 3 beta, okay? And when then glycogen synthase kinase 3 beta is phosphorylated, it will be inactive. Now it's inactive and not able to phosphorylate the glycogen synthase or inactivating it. So the glycogen synthase is remain active due to phosphorylation of the glycogen synthase 3b. At the same time, it will not able to phosphorylate and thus activating the glycogen phosphorylase. The ACET disrupted this reaction, and in this way again increase the activity of the glycogen synthase and decrease the activity of glycogen phosphorylase. That's all about the signal pathways for affecting the glucose uptake in the cells, also glycogen metabolism and lipid metabolism. Now let's move on to the second part of the lecture, which is the insulin as a growth factor. Okay, let's start. Okay, we said the insulin is bound to somewhere between the two alpha subunits, and this bound insulin to the receptor leads to a conformation change in the beta subunit and activation of the tyrosine kinase, and then phosphorylating the beta subunit. Phosphorylated beta subunits recruits other proteins including the IRS type 1 to 4, and then the IRS is also phosphorylated by the tyrosine kinase and bind to other proteins. One of them is PI3 kinase through a domain which is called SH2 domain. What's SH2 domain? Sarc homology 2, because it is similar to Sarc protein, short name for sarcoma. Okay, the PI3K converts PIP2 into PIP3, and then PIP3 bind to PDK type 1 or 2, and also protein kinase B or ACET. Protein kinase B is activating what? mTOR, mammalian target of rapamycin, sometimes called mechanistic target of rapamycin. mTOR then activating or affecting other proteins. One of them is called what? One of them is called eukaryotic initiation factor 4E, and also other protein is called eukaryotic elongation factor. Okay, mTOR activating two other proteins. One of them is called eukaryotic initiation factor 4E, and the other one is eukaryotic elongation factor. Sorry. Okay. At the same time, the ACT protein will phosphorylate the glycogen synthase kinase, right? The glycogen synthase kinase type 3 beta is phosphorylated, and once phosphorylated, it will be inactive, while the mammalian mTOR is active and is phosphorylated. Glycogen synthase kinase is responsible to inhibit another protein which is called what? This protein is called eukaryotic initiation factor 2B. The mTOR activating the EIF4E, while the GSK3 beta in activating the EIF2B. What are these proteins? From the name, eukaryotic initiation factor and eukaryotic elongation factor, these are involved in affecting the translation, initiation, elongation of what? Of translation of proteins. This is what? Translation control. The translation is composed of what? Initiation, elongation, and termination. These proteins are involved in increasing the rate of initiation and also elongation of what? Of translation. The second part, or the other side of the IRS, will bind to another protein. This protein come to here. Again, through what? Through the SH2 domain. SH2 stands for what? Not sulfhydryl, it is sarcomology 2. This protein is named what? GRB2. Growth hormone receptor bound protein 2. Okay? GRB2. GRB2 has two hands. Okay? This one. And this is the okay. The GRB2 has two hands. One of them is what? Sarcomology 2, which is bound to what? To IRS. Sarcomology 2. And the other two ones are Sarcomology 3. Okay? Now, when the GRB2 is bound to the IRS, it will be active and bind to other proteins. So it's not an enzyme, actually it's another adapted protein which links the IRS to other proteins. Which proteins come to here? The protein names SOS. Okay, this is the SOS. Okay, SOS stands for what? Sons of sevenlet, which catched by the two SH3 domains, like two hands catching the SOS, son of sevenlets. Subunits in turn is activating another protein which is called what? It's called RAS. This is the RAS, stands for what? Rat sarcoma. Okay. This RAS is active when it is bound to what? GTP, guanosine triphosphate. When there is GTP, it will be what? It will be active. But when it's bound to the GTP, it's inactive. What's the role of SOS? SOS. SOS is a guanosine exchange factor. It means removing any GDP on the RAS protein into what? Into GTP. Exchange any GDP into GTP. And thus the RAS will be what? Will be active. For example, you have a GDP. This SOS is a song responsible for what? Responsible for exchanging any GDP into what? Into GTP. So the RAS is what? Is active. RAS in turn activate another kinase protein which is called RAF. Rapidly accelerated fibrosarcoma. RAF is here, is now active due to interaction with what? With the RAS protein. RAS converting or affecting another enzyme which is called MEK. MEK converted by RAF, usually it's RAF1 or cellular RAF, converting into another protein or enzyme in the state of phosphorylated. Okay, this MEK is inactive when it's not phosphorylated, but by adding a phosphate group to it, it will be active. MEK in turn affecting another protein kinase, which is called what? ERK. Again, ERK is inactive when not phosphorylated, but by phosphorylation, it will be active. And the ERK affecting or phosphorylating another protein. Let's say this is a nucleus. This ERK activating a protein which is called ELK. Okay, ELK usually type 1. ELK is activated by phosphorylation. So adding a phosphate to here, it will be activated. By which enzyme? By the ERK enzyme. So the ERK activating the ELK1 by phosphorylation. ELK1 will associate with another protein which is called SRF. SRF stands for what? It stands for serum responsive factor, serum response factor. Okay, now both are bound to each other and the SRF is active. SRF in turn is responsible for what? It is a transcriptional factor. Okay, so leads to the transcription of many genes. Again, the IRS will recruit another protein which is called GRB2, growth receptor bound protein 2. This has one SH2 domain, sarcomology 2 domain, and 2 SH3 domain. Through the SH3 domains, it will bind to the SOS protein. SOS protein is a guanosine exchange factor. Guanosine exchange factor. So exchanging any GDP into GTP on the RAS protein, which is a membrane-bound protein. RAS, when bound to the GTP, activate the RAF1 or cellular RAF, okay? RAF1 or cellular RAF, and RAF1 in turn activating the MEK by phosphorylation, and MEK phosphorylating the ERK, and ergos phosphorylatase is active and able to phosphorylating the ELK1, and ELK1 will, which is a transcription co-activator, will bind to the SRF, serum response factor, and leads to the transcription of many genes, including the genes responsible for cell division. Because these genes are responsible for cell division, they are called what? Mitogenes. So this ERK1 is, we can say, belongs to a family of proteins called what? Called MAP kinase. MAPK. Why MAPK? Mitogene activated protein, because this protein, which is a mitogene activated protein, is activated by what? By a mitogene. Insulin is a mitogene. Mitogene means any peptide or molecule which increase the or induce the cell division, mitosis. Okay, so you can say it belongs to a larger family of what? MAP kinase, because it's phosphorylates a MAP protein, which is what? ELK1. If this is MAPK, what's the name for this? MEK. Okay, it will be what? It will be MAP KK, means MAP kinase, phosphorylating the MAP kinase. And now this RAF is had another name, which is called MAP KK, MAP kinase, because it phosphorylating and activating the MAPKK, and in turn MAPKK phosphorylating and activating the MAPK. Okay, these proteins have two names: the RAF or MAPKK, MEK or MAPKK, and also ERK, which is MAPK. Now, through these two pathways, which are, one of them is PI3 and ACT pathway, insulin increase protein synthesis. At the same time, by activating the GRB2 and the MAP kinase pathway. This is called the MAP kinase pathway. Insulin increases the transcription of many genes. About 100 genes are affected by the serum responsible factor in response to activation of this receptor by insulin. Now, one more thing is remaining to discuss, is that every beginning has an end, right? This insulin action should be ended. How we can end it? The mTOR, mammalian target of rapamycin, and also S6K. These are two enzymes which are responsible for adding phosphate groups into the serine and threonine residues on other proteins. They're also able to phosphorylating the IRS, okay, as serine residues, and phosphorylation of IRS as serine residues in activating the IRS and leads to internalization of the receptor, and the insulin action will be ended. Okay, IRS was activated by adding phosphate groups into the tyrosine residues, but now by adding phosphate groups into the serine residues, it will be internalized. Basha, sorry, okay. But now by adding phosphate groups into the serine, it will be internalized. And another thing which is there to reverse this reaction, the PI3K converting PIP2 into PIP3. But there's a protein, an enzyme, okay, this enzyme is converting the PIP3 into PIP2 back, and the name for it is what? Or simply PTEN, phosphate and tension homolog. phosphate and tension homolog and converting PIP3 back into PIP2, and the protein kinase B will not be more activated, and these pathways and the pathways for the metabolism of the glycogen will be what? Inhibited. That's all about the insulin signaling and the cascades of other pathways to alter the function of the metabolic machinery, of the gene transcription, and the membrane permeability. In the next lectures, we can talk about the insulin actions in different tissues, most importantly the liver, the muscle, and also the adipose tissue, and also then other tissues. Thanks.